Great, thank you for coming. Okay, we come from Valencia, Spain, and our project is for tested cellular biosensor, making E. coli testing flavor. The outline of this talk is an introduction, and then we are talking about design, part design, and system design. Then we present some experimental work and some conclusion and final. So the objectives of this talk is to obtain a modular system. So we design different devices. So also we want to obtain a circuit with few genes. So the target behavior we search is a gradient response according to a given concentration of an input. And for that, we use a biological pathway in order to connect that device, those devices. And finally, we use new synthetic parts. Okay, we design a sensor system and an actuator system, that is to say, a genetic network. So we use OmpR as intermediate mediator in order to connect those systems. So we can see the input in our case is a given molecule. We have chosen a vanillin molecule. And this module has the output of OmpR, and the actuator has that protein in order to activate using OmpR, which takes that protein as a transduction factor. And we use some fluorescent protein as a. So our circuit is based in the waste work, and we obtain an incoherent system, and the interpretation is semi-digital, that is to say, we don't consider a Boolean explanation in order to explain the circuit. So we need a little bit more complex. So this is the actuator behavior we want to achieve. So if we increase the vanillin concentration, we achieve some different colors. If we know vanillin, so the color is gray, and at intermediate levels we have a red color, and high levels of that molecule we have green color. So our vanillin receptor is a PBP, that is to say a periplasmic binding protein. So that PBP sends the vanillin molecule and frees a phosphorus, and that phosphorus phosphorylate that OmpR transcription factor in order to activate our genetic circuit. So we use a computational procedure in order to design that protein. So that protein, that periplasmic protein, has two conformations. The first one is open and has high stability. But when a vanillin molecule is close to that conformation, it's more stable if the protein cuts the vanillin molecule and the conformation is closed. So in this picture we can see the affinity of this design. We can see the bindings between the ligand and the receptor. So we use a sensor of ribose in order to change the affinity. We change the affinity to sense ribose to sense vanillin. So we have designed some parts. The first part we have designed is a promoter. So promoters are parts very interesting in order to obtain a given response. So we bet to binary promoter, that is to say promoter with more than one transcription factor. So we use two transcription factors in order to regulate our genes. So with this kind of promoters, we can integrate two signals. For instance, if we want we have like and C1 with those promoters, we can integrate two signals. And in a small in a small size, we have a very powerful device. And we can achieve different implementations, for instance, to repress or to activate one repressor and one activator. And then we have designed that protein. So our methodology is based in a combinatory procedure. So we use design as a bioinformatics tool to design proteins. So we use the inverse folding in order to design the protein. We use that expression of the energy, considered vulnerable, electrostatic, and solar tissue. And the combinatory problem is to take into account other possibilities. So each amino acid or each rotamer has many conformations. So the average is 10. And so if we have 20 amino acids, we have for a chain with n amino acids, we have 2,000 n possibilities. So we set in the phase space the best conformation. So there are two criteria: the binding criteria and the stability criteria. So these two criteria vary. So if we want to achieve a very stable protein, we reduce the binding. And we want to achieve a high affinity, we compromise the stability. So we take a solution from these Paretos from a point from that base. So some words about system design. Our implementation is using an incoherent system. We use two branches, a positive branch and a negative, using an inverter. And we use some well-known proteins in order to implement that: CRP, beta, C1, DFP. So if we have low vanillin concentration, we have very few phosphorus, and this promoter is not activated. So CRP and beta are not expressed and we don't have any color. However, if we play with the pure PR promoters, we can do a system that at intermediate vanillin concentration we can activate only the first one branch, activate expressing CRP, and we obtain CRP and C1, and we obtain a red receptor. In the final case, we obtain only CRP but not C1, and we obtain GFP according to this picture. So some words about model and simulation. So we consider an effective model is to say we model only protein concentration, not mRNA or other species. So we take genetic parts. So we use nominal parameters taken from literature, and we perform our simulation. So we obtain that response. So for the GFP, we obtain this yes gate, that is to say, is more or less proportional. And for the GFP, we obtain a pulse. So at intermediate concentration, we have this response. And if we superpose that columns, we can obtain a derived column. We perform an analysis, a sensitivity analysis, in order to study the robustness of the system, because we use genetic parts in our simulation. So we present the evolution of red protein versus OmpR. So we can see that more or less the expected behavior is good. We make also a robustness analysis in order to study the input signal. So We introduce a noise, a white noise in OmpR. It's the same thing to introduce a noise in the vanillin concentration. So taking into account some interferences or things like that. And we see that our system is very robust versus that noise.
So once we had simulated and studied the circuit in silico, we had to take all those parts into real world and see what we needed and where were they. So what we did first was to analyze how would we do the sensor is the yellow part and the actuator is the light green one. And we thought that we needed the periplasmic binding protein to vanillin, then the fusion protein, and the circuit with the TET-R inverter, and the OmpR, that are the promoters regulated by OmpR phosphorylated. But we needed the native one and the mutated one, that is the one with the TET-R and with the GFP. And then the AND promoter, or the synergistically activated promoter with the RFP. Okay. So we found the parts. We found the parts, most of them at the repository. We had like 80% of them. But we had a little bit of problem with those two because they had to be taken from the E. coli genome. So we did some genomic PCR in order to have them. And then we had to make it biobrick, as we will see later. And then we built from scratch 2 parts as the AND promoter and the vanillin periplasmic binding promoter. Okay, we'll now see a little bit how we did experimental work, how we managed to get those parts and made it in biobrick. The AND promoter was a published one in the 94, and what we did, we constructed it from scratch. We took the PRM promoter from the repository. It's a C1 lambda C1 activated promoter, and we thought of adding upstream some CRP binding site. The problem with the CRP binding site is that it located XBA1 restriction site. So we studied, the mutagenesis of that, but we found that the binding affinity would be much more lower if we mutated it. So we did it like that. We ordered 11 oligonucleotides, and we did the DNA synthesis with thermal cycles on the PCR machine. We ligated with ligase, and then taking the extreme oligonucleotide, we made some PCR in order to have that amplified. Okay, the vanillin receptor was in silico design with the designer program. We looked at the 14 mutations that had to be done on the pocket that binds the vanillin, and the amino acids were optimized for use it in order to use it on the E. coli chassis. Okay. Then the fusion protein was also a published one. What we needed was the periplasmic and transmembrane domain of the TRG, and only the EnvZ, the cytoplasmic domain of the EnvZ. TAR EnvZ is also a part of the registry, but we didn't want the TAR. We wanted the periplasmic domain of the TRG. So what we did was amplify both, digest them, and dephosphorylate the TAR EnvZ in order to blow the relegation, and then we mix and ligate. And then we amplified with one primer of the TRG and the reverse one of the EnvZ in order to have much more good fusion protein or interesting fusion protein than the TAR EnvZ one. Okay, and then we wanted to change the backbone model TRG and EnvZ in order to make it a plasmid with ampicillin resistance. We just digested it with EcoR1 and PST1 with the phosphorylated vector. And then we mix, ligate, and transform. The PTET RFP is very interesting because only on the plate, on the petri dish, you get red colonies. It's something that doesn't happen much with the GFP. So that was interesting because we knew that the red colonies were a relevation of the biobrick, and the white ones were our biobrick. Okay. Then we went to the FACS in order to make some analysis. We used FACS for two main purposes. One of them was clone identification, fluorescent clone identification, obviously. We used a negative control like normal strain, like XL1 blue, so as top 10, or then we used a positive control, typically a green fluorophore, and the different clones we wanted to identify. Those of them that passed through the threshold of the negative control were good for us. And more interestingly, we used also the FACS in order to make a rough promoter characterization. Here, there's the characterization of the OmpR and the mutated one. Okay. The black one is the mutated, and the green is the wild type. This is not a very accurate statistical analysis, but it helps us in order to see which one of them is stronger as a relative comparison. Okay. And finally, the parts we submitted were the ones we want to talk about. Note that the very interesting part of the fusion protein is that it's good enough in order to make some modular detection systems, because all the periplasmic binding proteins bind to the TRG, to the periplasmic part of the domain of the TRG. So you could mutate, as we did, the pocket of the periplasmic binding protein in order to detect some given molecule. I'm thinking, for example, of the MIT project of this year, the banana scent. The isoamyl alcohol could be detected, if you work on that, could be detected with some periplasmic binding protein, and then you could use a TRG and EnvZ fusion protein. Okay. Then conclusions. We have designed a 7 genes genetic network that gives us a graded response according to a given interesting molecule. We use the phosphorylation mechanism in order to connect the sensor part and the actuator part. So that OmpR gives us the bridge between those modular parts. And then the interesting part of the AND promoter is that you can reduce the number of genes and then integrate branches of your network. And then you can directly mutate the PVP in order to detect whatever you are interested in. So acknowledgements. We're very thankful to the Symbiocom project, asked for our universities and the scientific guys that helped us out with the lots of problems we had. And so that's it. We are two making the talk, but let's say that our group is a 17, 18 people group, so we are pretty one-shot guys.
Right, thank you. We have 6 minutes for questions. And let me just ask you first, which was your favorite part and why did you like it so much?
Vanillin receptor.
And why do you like that one?
Sorry?
Why is that your favorite part?
Okay. Because it's a powerful part in order to design this synthetic part. So we use a computational procedure. So I think it's a powerful methodology in order to design new part. That in nature, for instance, it doesn't exist a protein level. So we can design new proteins in nature.
Right.
It doesn't appear.
Okay. Any other questions? Yeah.
I wanted to ask why you chose CRP as one of your dual regulators, because it has so many connections to the investors.
Yeah, I think so. But PM promoter is based on young work. So this is that group used C1 and CRP as inducers. So we only reproduce that work. So, but we think that CRP interact with the genome and pathways. So, but we think that the interference with our circuit we expect that will be reduced because the amount of CRP may be low in order to interact highly with the network.
So how far did you get in the project? What have you actually done and not done?
Yeah.
Let's take a look at them. So we have the vanillin PBP, we have the fusion protein, as we have that for the sensor and then for the actuator. The only one that we don't have is the CRP. Let's say that maybe if we did the Jamboree on 20 December, we could have tried to ligate all of that and put it on one single equal chassis. It was a time problem, really, because the parts are already on our fridge. Except
Have you done some other readout without the green part, but with the yellow part intact in cells, showing that it can just produce anything, just a single signal?
No, not yet. But we will work on that.
We had many problems with issues.
What was your greatest challenge, do you think, that you overcame?
Sorry?
What was your greatest challenge that you overcame?
The greatest one was, let's say, a week, one single week we got, so the ligations didn't work for a protocol problem, and then when it started working, we had the chemioporating cells were waste and were infested. So when one worked, the other didn't. So that was a little bit of a crash. But we can.
We have time for another question or two. Yeah.
So how easy do you think it would be to if you wanted to detect different protein? Basically, your procedure for designing this protein, how generally modular is it?
I think we use a computational procedure in order to design periplasmic binding protein in order to detect, for instance, a given protein. A given protein. So if we use that sensor as a module, you can use OmpR in order to activate another module. So if you use a genetic network that takes OmpR as an input, you can use our system, our sensor system. So you have a little bit of modularity.
There are some works on TNT detection that they use the periplasmic binding protein and they use a fusion protein like TRD and EnvZ, and they get OmpR phosphorylated. But the thing is that they only put a pure OmpR with GFP. So when you detect the TNT, it blew green. So I think it's a quite powerful modular ability for that.
Okay, let's thank our speakers again. Next is Bangalore.